guess we'll get started with today's material. So are there any questions about anything, any issues yet from the past few classes before we get going? You're all up to speed on what gene expression is, how you measure it, and how you do computational analysis on it? Okay, then I guess what we'll be looking at today is some of the direct data resources we will be using, some of the ways we can look up gene expression data. In some cases, that will be raw data from the direct expression studies on which we'll want to do analyses. In some cases, we'll want to be able to learn about what's already been learned from other studies. And we'll see examples of a few of these things, both raw expression data and information about already known patterns of expression. And to do that, I'm just going to jump straight into some of the things we can get to with web browser. I'm going to start with jQuery, as we've done many times before. So we're going to start by doing a jQuery search to learn about some of the resources we can get access to through the NCBI. As is the case with many of the kinds of resources we're looking at, the NCBI has some really good data available, really good repository, and it probably has the best kind of central repository for gene expression data, what's called the Geo or Gene Expression Omnibus repository. And so I'm going to start by looking at that, by imagining that we are interested in a particular topic of study. So we talked last time about some applications of gene expression, and we used some examples using a data set looking at expression in cancers, particularly in lung cancers. And so what I want to do at this point is introduce how we would go about accessing a data set like that and learning about some questions related to lung cancer development. So what I'm going to do is type in a search here. Let's say lung cancer. And maybe we're specifically interested in risk factors for lung cancer, and the most important risk factor would be smoking. I'm going to type in this query, lung cancer smoking, see what comes up. And as we might expect, that's going to get hits to a lot of different data sets here. So we're going to get some hits to literature data sets. There are quite a lot of scientific papers out about connections between lung cancer and smoking. But we're also going to get some hits to these Geo repositories here, which are the ones that, as I mentioned, refer to gene expression, the Gene Expression Omnibus, and that is divided into two different resources that we're going to be accessing here. The Geo Data Sets is the main one we'll be interested in. The Geo Data Sets repository is basically collecting complete studies as well as other repositories of information that we care about when looking at these kinds of topics. GeoProfiles is collecting expression data on particular genes, and we'll see that these are tightly linked together when we get into some of the Geo data sets. So Geo data sets entries come in a few different forms, but the ones we care about generally are going to be the ones actually labeled data sets here. And if we just click on this tab, data sets, we can reduce it to a relatively small number of hits to complete studies that match this query, lung cancer smoking. So we can see that some of these are kind of not quite what we're looking for. This first one is specifically looking at lung cancer gene expression in non-smokers. It's not the question we care about here. In some of them, we may have things that are kind of on point for what we're trying to do. This is looking at how smoking specifically affects gene expression in the nasal epithelium. And there are various others that may be related or unrelated to what we're trying to do. I'm going to scroll down to one of the older ones down here. So at number 10, one that's looking at how smoking affects gene expression in the airway. So looking at the airway transcriptome. And I will click on that, and that will bring up one of these Geo data set entries. So this is going to be a page devoted specifically to this one study. And there's a lot of information we can get here that we would want to look into if we were thinking about maybe following up on this data set to learn about the activity of our particular genes. One of the main things we often care about when looking at a study like this is how it was conducted. So what was the technology for assaying expression? Can anyone figure out from looking at this here what kind of technology was used to study the expression in this particular study? Well, we can see there's this term here, platform, and next to this we have something describing an Affymetric human genome U133A array. So array is going to be the key here that will tell us us that we're looking at a microarray study, most likely. And in this case, it happens that this Affymetrix Human Genome 133, or A array, is one of the standard kind of human genome or human expression arrays that you could order if you wanted to study patterns of expression in humans in particular. So there are catalogs you can go to order these. This is an older one that's been around for a while because this is an older study in part. But Affymetrix is one of the companies that makes these, and this was a study conducted in particular with one of their arrays. And we could go to this GPL 96 link here if we wanted to learn all about that particular platform. So another kind of entry that you find in these databases is information on the platforms that are used, some of these standard methods often. And we can see about other studies that have used this particular set of arrays. So there are over 1,000 different entries in this Geo omnibus that use this particular kind of 133 array, and we could get detailed information if we wanted it on exactly what is on this array. This particular one contains about 22,000 distinct spots, and if we wanted to know what those spots were, we could download a complete table of information telling us exactly what gene was profiled on each of these given spots, what is known about the behavior of that gene in some cases, of various kinds of annotation, but basically figure out how to translate these spots into the information we care about. So that's one thing that we would care about when looking at these studies. What was the technology used to do the study? Can anyone tell me what useful information we get out of knowing that this is a microarray study? How should that inform our analysis of this particular data set? What are the characteristics of a microarray study? Yeah.
It's a little bit less accurate than real-time PCR,
Yeah.
some other type of assay.
Yeah, so microarrays tend not to be a very accurate assay. They have a very high noise level, so that means it's often hard to find significant differences unless you have a lot of replicates, so a lot of samples, or do other kinds of corrections to make sure you can get a good signal. They do tend to have a fairly large number of genes profiled. In this case, I mentioned just over 22,000 spots on the array, so you can do more or less full genome kinds of studies with these. But that is basically what we get out of looking at this particular array. And what we can then do is go back to our data set here and ask about some other things we might care about if we were looking at this particular study. So one of the other things we would often care about once we've determined what spots are here, so what things are being profiled in this array, is what samples are present. So who was studied, or what samples were studied in this particular assay. If we really wanted to know the details, you would often find a citation, so a scientific paper published about a study like this, and you could go there and read in great detail about what samples they used and why they used them and how they prepared them. So often this is something we would want to look up if we were really using this study in a serious sense, but for now we can go to kind of a summary by clicking this sample subsets tab here, and that's going to tell us a bit about what samples are on this particular array and what distinguishes the samples. So what variables were they looking at when they tried to see what might affect expression in this study. And this is a study specifically on the effects of smoking on gene expression in the lung. So it's not surprising that we've got one category of samples that consist of people who smoke. So we've got current smokers here, and then each of those current smokers is identified with a unique label, and we get sample IDs. So there is a web page devoted to each of these samples that can tell us more about those particular individuals. We also have a set of samples of people who have never smoked. So obviously you want to control if you're looking at how smoking affects your expression. And we also have some former smokers here. So some individuals who smoked at one point and stopped smoking. So that's telling us basically what's in our data set. And that is kind of a preliminary we can use if we want to start asking questions about what this data set can tell us about the effect of smoking on gene expression in the lung. So are there any questions yet at this point? Okay, so these tend to be relatively large data sets, and there's a lot of information we can get out of this. The Geo Profiles data set is linked to this Geo Data Sets one, and we can get access to the individual profiles, which are the patterns of activity for single genes assayed in this study. So if we go to that Expression Profiles tab, we can see that there are entries in the Expression Profiles data set for each of the spots on that particular array in that particular study. So this is showing us, for example, an entry devoted to one particular spot related to a gene called PIR, or purine. And we can click on this kind of icon here to see how that gene behaved in this particular study. So we can kind of see that there's a pattern here. If you look at the red bars, those are the things that are kind of giving you the relative expression level across these different samples. And we can see that you have relatively high expression of the samples that are categorized with this light green, which is the current smoker category. And if we scroll a bit here, we can see that expression is quite a bit lower in this gene for people who have never smoked. So this is suggesting that probably something about this gene is causing upregulate, or something about smoking is causing upregulation of this particular gene. The former smokers, you might note, have kind of an intermediate pattern. Some look indistinguishable from the people who have never smoked. Some have patterns that look more similar to the smokers. That may mean that it takes a while for the effect of this of smoking on this gene to go away. Maybe it's a lingering effect that takes some time. It may be that some of these people are actually not being honest about being former smokers. Maybe they're still smoking and it's showing up in the gene expression pattern. You always have to be aware that that kind of issue comes up when you're looking at human data. But basically, this is showing us what a profile of one particular gene looks like. And if we wanted to look in more detail at that, we could scroll down here and see exactly what the list of values were for each of these subjects in our study for this particular gene. And we would have a page like this for every gene, so every one of those 22,000 spots that was profiled on this array. So any questions about that? Okay, if we really wanted to know what the relevance of this gene was, of course this is linked to other NCBI resources. We could click on this PIR link here, get an Entree gene page for that gene, use that to start following up on what the function of this gene is, what literature is associated with it, if there are diseases associated with mutations in this gene. So all the things we've been seeing in previous weeks, we can get to fairly easily just by following through these links. But what I want to do is really get into how we use some of our microarray or gene expression study specific analysis tools by going back to this particular page and looking at some of the ways we can computationally analyze this kind of data. Now, one of the things we would often care about in a study like this, if we're trying to ask what smoking does to gene expression, is we would want to ask the question, what genes change their expression when someone smokes? So what kind of tool would we use to do that? What's the analysis method you would commonly use to ask a question like that? In this case, we know what our categories are. We know there's some people in our data set who are smokers, some who are non-smokers. We want to go through individual genes and ask, is there a significant difference in the expression of the gene in smokers versus non-smokers? So how would you ask that? Well, we had seen last time that that's something we can pose a statistical hypothesis testing question. In particular, typically the statistic we would use for that would be a t-test. And this particular resource has some of these basic analyses built into the pages, so we can just access them directly. I will say, usually if you were doing this on real data sets, we're actually trying to study this question, what you would want to do is download the data set and put it into some offline analysis tool that's much more fully functioned than this. It has a lot more options, but this lets you do some basic kinds of tests right inside the browser, so it's a good thing to use to kind of play around with the kinds of tests we might want to do. And one of the kinds of tests we can do here is to compare two sets of samples. So I'm going to click on that, and that's going to give us a dialog here that lets us walk through how we would analyze whether there are statistically significant differences in any gene between these two samples. This doesn't have anything built in for more than 2 samples. So that's one application where we might, more than 2 sets of samples. That's one application where we might need to go to a different kind of tool. So MATLAB or R, or the specialized gene expression tools. So what kind of statistical test would we use if we were trying to compare 3 categories? Like if we were trying to find things that distinguish between current smokers, never smoked, and former smokers? So that you would commonly use an ANOVA test. So ANOVA is what you would use for more than 2 categories. T-tested, it's exactly 2 categories. But here we'll stick with 2 categories because that's what is built in here. One thing we would need to do is pick the samples. What I'm going to say here is we will take, see this group here, which is the current smokers, and I will assign them to category A. So just selected the current smokers, and I'll assign the never smoked to category B. So now they are assigned to group B, and we'll just ignore the former smokers here. That seemed like maybe a noisy signal, so we're just going to say we're not going to pay attention to them, and we'll just try to compare current smokers to never smoked. So we'll click on that, and then this is telling us exactly which samples from our data set fall into each of those 2 categories, and we then want to pick a test to compare those 2 categories. So we have a few options here. Usually the thing we care about is a t-test. So if we want to know about genes that are either overexpressed in smokers or overexpressed in non-smokers, that is underexpressed in smokers, then we would want a 2-tailed t-test. So that's going to test for effects in either direction, so elevated in smokers or elevated in non-smokers. You could get that information by doing one-sided tests, but doing that twice, usually that's not going to be the right thing to do if you're looking for a 2-sided test. Suppose we were interested only in genes overexpressed in smokers, though, so genes whose activity goes up in smokers. Why would we want to use a 1-tailed test for that rather than doing the 2-tailed test and just throwing out the ones that are in the wrong direction? For any given magnitude of effect, the p-value is going to be larger, so less significant for a 2-tailed test than for a 1-tailed test, because you're considering both the effect in the positive direction and in the negative direction when deciding things that, basically when deciding what the chance is that we have a stronger effect just by random chance. So if you're really only looking in one direction, you want the 1-tailed test because it will be easier to find weak effects. So let's leave that there. And I will change the significance level as low as it goes to 0.010. So one unfortunate thing about this tool is that this doesn't have multiple hypothesis correction built in. So really, if we were looking at an array this big, we would want a much lower significance level than this. We want something that is Bonferroni correcting for the fact that we're looking at 22,000 samples. But for here, we'll just use this, and we'll just have to assume that later on we would do other validation to decide which of these are actually significant. But we can leave that there, and we can query group A versus group B. And what this should do is bring up a set of results, so it's that set of the gene profiles that it's saying have a significantly different expression level in those 2 samples. I'm going to click on this thing here that says sorted by default order and change that to subgroup effect. So that's going to group these basically by how strong the effect is. It's often going to be the thing we care the most about. We can see that that PIR gene comes up as the top effect here. And you can tell from this icon, which is a little version of that expression plot I was showing you before, that this is a gene that has high expression in the current smokers, low expression in the never smoker people, as we saw before. We also have some other results here. So the next 2 genes are kind of interesting. We see a gene CYP1B1 and CYP1A1. So these are 2 different genes, but they're closely related genes. They are both describe the pattern of expression effects on these genes. How would you characterize how expression is affected by smoking? Well, these are genes that we can see. If we click through here, we would get the labels, but remember the labels from before. Light green is the current smokers, medium green is the never smoked, dark green is the former smokers. So we can see that these are both genes that have greatly elevated expression in smokers versus never smoked people, and again, kind of intermediate in the former smokers. So again, it could be that some of these former smokers are not being honest and they're still smoking. Could be that it just takes some time for the effect on this gene to go away. But both of these are genes that are upregulated by smoking. In this case, I say it's kind of interesting because these cytochrome P450 genes are genes that are involved in breaking down small molecules. And one of their very important functions is that they clear toxins from the body. So it's maybe not surprising that you see these popping up very strongly in the study of smoking. If you're smoking, you're introducing a lot of toxins to the lung tissue. These genes are turning on as part of a module of genes that clears that effect, or is trying to clear the toxins from the lung tissue. So that's why we see that effect here. So if we scroll down a little more, we can see that number 5 here, CYP1B1, is actually the same as number 2. So our 5th highest result is actually the same gene as our 2nd highest result. Can anyone think of why that's coming up twice? Why do we see 2 different results for that gene? Well, the important thing to remember is that what this is testing is not really genes. It's testing spots on the array. And it apparently just happens that they have 2 different spots on the array for this particular gene. It might be because there are splice differences and they're trying to put these spots of the antisense DNA strands in different points in the gene to catch different splice forms. It might just be that they know this is an important gene and they're trying to have some redundancy so we can maybe average between those to get a more accurate profile of expression. But for whatever reason, they have 2 different spots. But we can see that those spots show very qualitatively similar patterns. It is 2 different isoforms. They show similar changes with respect to expressions. So any questions about any of that? Okay, so that basically is showing what we can do with t-tests here. And again, really, if you were trying to do a study like this, you would want to download your complete data set and do some of this offline in a more fully functioning either gene expression analysis toolkit or a statistical package. So you can do more sophisticated tests. You can get your von Fermi correction right and so forth. But that gives you a way of doing some of these tests in at least the basic version. Suppose we want to ask a different question, though. So suppose we were interested in whether there were modules of genes that behave differently across these samples for reasons unrelated to smoking. In this case, we don't have a lot of information about the samples in our data set. We know which ones were smokers and which weren't smokers, but we don't have any labels that tell us whether there are any other meaningful categories here. So if we wanted to discover categories like that, what kind of tool would we use? We wanted to discover a category that's not already labeled in our data. That's something we would commonly do by clustering, and last time we learned about a couple of ways of doing clustering, and versions of those are built in here at this cluster heat maps tab. So I can click on this, and this is going to give us a few different options for clustering our data set. We can see that there is hierarchical clustering. That's one of the versions we learned about. And what they're referring to as partitional, which the k-means clustering algorithm we learned about last time is one example. So we have those kinds of analysis tools available. You can see what we would get out of this by maybe starting with the hierarchical clustering. I'll maybe leave this on uncentered correlation. So that is saying that we will measure the distance between different samples or between different genes based on the correlation coefficient between the patterns across samples or genes, which is a statistical way of saying how similar they are. And the linkage refers to how we assess difference between subsets of genes or samples. So I will make this average UPGMA. So So average UPGMA is the way I explained the algorithm last time. It basically says if we're trying to measure the distance between one subcluster and another, you take the average expression pattern in each subcluster and use the distance between the averages to measure distance. So we can click display here, and that will bring up a hierarchical clustering, where we can see here we've got a clustering of our samples as well as a clustering of our genes here. Now, one of the things I mentioned before is that usually by default, the color code is that something that is higher in the sample you're measuring will be colored red or magenta in this case. Something higher in the control will be colored green. They pick those colors because that happens to be approximately the colors of the actual fluorescent probes people use, but it's often helpful to be able to change these. So I will pick colors here, and maybe we will say that high expression will be yellow and low expression will be blue. It's usually a good idea to change these things because anyone who is colorblind will have trouble seeing red and greens. It's just unfortunate that that's kind of the standard, but it's easy to change. And then we can pick out more clearly some of the patterns of variability here. So you should be able to kind of see that there is a pattern of genes with low expression, or excuse me, samples that have low expression, a subset of genes here. This doesn't come through as clearly on the screen as it looks on my screen here, but we can also see there's a module of genes with high expression, a subset of samples here. We could look in more detail at exactly what's going on there, and we can kind of see how the hierarchical clustering of genes is picking out some of these subgroups. So we can see a very dense subtree here that's capturing a large group of genes that have this pattern of high expression in these samples, low expression in these samples, maybe medium expression in these samples. If we want to get more detail on that, there are tools built in here that will let us look in more depth here. We can kind of just click on this to pull up a dialog that lets, or a box that lets us select a subset of genes. We can click on that, and we can bring up a more detailed view of this particular set of genes, find out exactly what genes are popping up with that pattern, and if we want to go in and investigate the behavior of those particular genes. And as with many of these things, this is nicely integrated into some of the other resources we have available. So we could go up here, click View in Entree, and that's going to bring up the profiles for those particular genes in this particular study, so we can get all the raw data, look through these, and see if there's anything particularly interesting to us in there. So any questions about any of that? Okay, so one reason we are often interested in these hierarchical clusterings is that sometimes we don't know the right level of resolution to look at, and there we can kind of see how these trees give us an idea of what the important subsets of samples are, or the important subsets of genes. So we can kind of see just from the tree that if you sort of cut it at this level here, we're picking out a set of submodules of genes that seem to be more or less acting the same with respect to the categories considered in these samples. That's one reason we often want a hierarchical clustering. We don't always know the right number of clusters for analysis, and the tree can help us guide that kind of decision. In some cases, though, we do want to go with a k-means clustering or something similar. So we can go back and pick this partitional clustering here, and that will give us a dialog that lets us do a k-means or other similar kinds of clustering measures. Maybe for this one, what I'll do is I'll set the coloring so high expression is red and low expression is blue, and we can pick some different options for how our k-means clustering works. We'll say that maybe the distance between genes that we're using to decide which genes are more or less similar is Euclidean distance. So that is what you would get if you plot them as points in a high-dimensional space and then ask what's the linear distance between these. We can pick mean here. That refers to the question of how we decide the representative of a cluster in the k-means algorithm we saw last time. And maybe we'll say that there are 3 clusters we're looking for. For this one, we have to specify that, and we can display a k-means clustering, and we can see what we get out of that analysis. So here we can see it's picked up 3 clusters, which is what we wanted here. We can see that this one has kind of a pattern of low expression in a subset of our different samples here. This is a set of about 7,000 genes with low expression in those samples. We've got kind of a mirror image cluster, same sets of samples popping up, but with high expression in these 4,500 genes, and then some that don't show a very strong pattern. We see kind of a mixture between these two. If we wanted to click on one of these, we could see what's going on there, what the particular samples are. And you can make out the color coding here. We can see again, these are our current smokers, these are the never smoked, and these are the former smokers. And we can see this pattern of activity of the genes with respect to those categories. Just shrink this a bit to bring that up more clearly. So how would you describe this pattern of expression with respect to these smoking categories? Is this clustering revealing a smoking-related subgrouping of our genes? In this case, it doesn't look like it. So there is a strong separation of our samples with respect to this cluster of genes. So some of them have very strong expression, some much weaker expression. But the clustering seems to have nothing to do with the smoking categories. Each of our three categories has a subset of samples with high expression, a subset with low expression. So this is picking out some strong effect here. It just happens not to be the effect they were looking for in this study. It's not obvious. We may not have the information we need to figure out why this effect is popping up. It could be a genuine biological difference. Maybe it has to do with the time of day they took the biopsy. Maybe there's a change in how they processed some samples that didn't occur to them would be important here. Maybe it's just a chance effect that they happened to run the samples on different days and their medium was a little staler one day or another, and that happened to overwhelm the change they were looking for. So as I mentioned last time, these arrays tend to be a very sensitive technology to that kind of technological artifact. And in this case, whatever it's picking out is not the smoking-related effects we're looking for. So we can get that from the t-test, where we know what the categories are, but in this case, the clustering is showing something else popping up as a stronger effect. Any questions about that? Okay, so that is kind of a run-through with one example data set of some of the things we can do if we want to learn about patterns of expression from a raw data set. But it's also important to know how to look up information that's already available about which genes are expressed in which tissues, at which times, in which organisms. And as is often the case, there is today just a ton of information available if you know how to find that. So I want to go through a few places we can get that kind of information. And this is one of the situations where often the best data you'll get is actually within model organism databases. Let me just close a few of these windows we've been opening and go to a database we should recognize, Mouse Genome Informatics resource. So Mouse Genome Informatics is one of these really good model organism resources. And just as it has great information about genes in mouse and mouse genome and mouse genetic variants, it also has great information about mouse gene expression. So we can see how that might work by going to this gene expression database here. Get rid of that. Click on Gene Expression Database, and this is going to have various ways we can access gene expression information specific to the mouse situation. And one of the things that's sometimes useful if you're talking about mice is this mouse developmental anatomy browser. So one of the reasons you tend to get good information from the model organism databases is that we often care about gene expression with respect to particular tissues or particular stages of development, and these are completely different if you're talking on one organism versus another. So maybe mice versus rats, you more or less have the same anatomy, but you really couldn't use the same tools to look at mice versus fruit flies, much less mice versus yeast. So we really would want different kinds of resources. So it happens that there is a nice categorization, what's called an ontology for mammalian tissues that's been applied to mice here. So we get this anatomical view here I may have to shrink this to show you in a bit more detail what this looks like, but if we shrink this a little, what we can see is that we have these different sorts of categories here that refer to different stages or different pieces, different anatomical components of a mouse. There's kind of a top-level category, which is just mouse. So mouse is the high-level thing that organizes all of the anatomy of a mouse. And then within that, we can find that there are subcategories for body fluids, body regions, body cavities. I'm going to scroll down to, say, organs. So maybe we're interested in particular mouse organs. And I can open brains. So mouse brains are very interesting. You can see there are lots of categories within mouse brains. Maybe we're interested in mouse brain stems. And we can keep drilling down like this to find whatever piece of mouse anatomy we're interested in. We can also, if we care about it, categorize that at particular stages of development. And just as the vocabulary is going to be different for pieces of anatomy in one organism versus another, there's a different vocabulary for stages of development. For mice, they use this feeler stage categorization. So this is describing different stages of the developing mouse embryo. And maybe we can see patterns of expression at feeler stage 15. We can also often, if we care about a particular kind of anatomy, just search for it. So you can say brainstem nucleus, and we can have that kind of pop up and show us the brainstem nucleus category, and then maybe say we care about expression in TS28, which is postnatal brainstem nucleus, and then we can look, we can see that a brainstem nucleus is a component of, we go up, a brain nucleus, and it itself has various subcomponents, so an abducin 6 nucleus, a cuneate nucleus, and so forth. But maybe this is the right level of abstraction for us. So we will click on this 602 here and bring up mouse brainstem nucleus expression results. So there are, as this says, just over 600 results on genes that have been profiled for expression, or at least assays of expression, done in the brainstem nucleus at feeler stage 28. And we can see all about the different kinds of results you can get here. So we see a lot of these are RNA in situ. That's a technique I didn't talk about. It's a way of testing for RNA expression in a tissue. So basically you fix a tissue and you kind of wash an antisense RNA or label DNA over your sample, and that's going to show you where your RNA is present within the tissue. So that's going to be a useful thing for getting this kind of information about where we do or don't have expression. And we can see that in some cases, this is going to report a positive result. So in some cases, we have detection in the nucleus, brainstem nucleus, or in some subpart of the brainstem nucleus. And in some cases, these are going to be reporting negative results. So I'm going to scroll down here to try to find one. All right, so here is one that is testing this gene ADCY1 and is reporting that this was not found to be expressed in the nervous system. You might notice there's a contradiction there, that this is reporting that it is not expressed in the nervous system. Other studies are reporting that it is expressed in parts of the nervous system. That's often the case with real data. Sometimes you see disagreement, and you have to decide what the balance of the evidence favors, or maybe go and gather your own evidence. We can see what kind of information is collected with these by picking one of these particular examples. So we'll take one of these RNA in situs. Let's take this one here for ADCY1, and I can show you what a page looks like for one particular assay in this data set if we really want to look at the evidence behind it ourselves. So we can see that this is a page devoted to this particular assay, so this particular experiment on this particular gene. Sometimes we will have image data associated with these, so we can see exactly where there is staining for the presence of this particular gene, and we can see exactly what they were testing and whether or not they found it present or not present, or in some cases say that it's strongly present at particular subsets of the anatomy and at particular stages in time. So in this case, they were looking at these particular pieces of brain anatomy, and in particular at TS28, and we can see that it is present in the places they were looking at that stage in time. Any questions about that? Okay, so We can also get this kind of information not just from the point of view of a tissue or a stage in time, we can get it from the point of view of a gene. So sometimes we have a gene of interest, and the question we want to ask is, where and when is that gene expressed? So we can click on the name of that gene or search for it, bring up information about other aspects of a gene. They will have information about expression from the point of view of that given gene. In this case, we can see that there are quite a lot of results. So tests for expression of this gene. We can see those broken down by tissues. We can see them broken down various other ways, but let's just click on the main result tab here, bring this up, and we can see about all the different studies, all the different assays that have been done that tell us about expression of this particular gene in various tissues. So we can see that at various points in time, in various tissues, this gene's expression has been tested, and we can see that in some of these it has not been detected, in others of these it has been detected. So often the results will be ambiguous. We may not have information for a specific result. We may have contradictory information, but there is a lot there, and if we really cared about this, we could look into that to find that information. It is worth noting that there are some other resources we sometimes want to use for this. So for example, if I go back here, one of the ones that's really useful if you're interested in expression, in particular in the neuroscience context, is this Allen Institute external resource. So this is something called the Allen Brain Atlas, which was a systematic attempt to study expression of pretty close to a whole mouse genome in mouse brains in very fine detail. And if we bring this up, we can see all the entries in this status resource devoted to this particular gene. So this gene has been studied 11 different times in 11 different brains in this resource. And we can click on one of these. And if we really wanted to get very fine scale information about where and when this is expressed in the brain, we can bring this up. We can, let's say, click on an image of interest to us. Maybe decide if this is a good one to pick. And pop up this image. And if we really want to know in very fine detail what the pattern of expression of this gene is, you can then go in here, zoom in on whatever fine detail of anatomy you care about, and really see at the cellular level exactly what kind of pattern of expression this gene exhibits in a mouse brain, let's say. So if you end up working in a neuroscience lab, this is a resource you might be using for your studies in that work. Any questions about any of that? Okay, so I just want to kind of quickly show you that similar sorts of resources are available for other model organisms. So we've seen quite a bit about fruit fly. That's another one you might work with. We pull up our Heartless gene we've been looking at quite a bit. We can see that this too has a lot of interesting information available on expression. So we get a tab here devoted to expression data of the Heartless gene. We can pull up that tab. We can see in a fly what stages and what tissues at each stage exhibit expression of that gene. We can see that for RNA. We can see it for protein expression, so immunological tests. And we can explore various high throughput data sets that have been developed to study specifically this question. So if we want to know about expression of the Heartless gene in fly development, we can pull up this modern code development RNA-seq data, and we can see exactly what the pattern of expression is found to be for fruit flies for this one particular gene. We can kind of see that it goes up. It has a peak of expression, roughly 6 to 14 hours during embryo development, drops down, has a second lower peak, relatively late in development, and then it kind of drops down again, so that after a few days after hatching, this basically has almost no expression. So this we would say is specifically a developmentally expressed gene, and of course we would have other ways we could look into where it's expressed, why it's expressed, what functions it has, and this too is often linked to external resources like image data, where we can find out exactly where the Heartless gene is expressed at various stages in development. So if you were working in fruit fly development, this again is a resource that is likely to be very useful for the questions you're asking. So any questions about any of that? Okay, so there are a few other things I wanted to show you in the remaining minutes of class. One of them I'll just show you very briefly is resources for protein expression. I've mentioned that usually when we look at expression, especially on a large scale, we're looking at DNA or RNA expression, just because it's easier to get the data on a large scale. But there are some ways of getting access to high throughput or at least medium throughput protein expression. One of these is a resource called Swiss 2D Page. So this is a collection of 2D page gels we could use to study expression of expression of proteins. We could click on the search by description. We can type a keyword here, say E. coli, and we can bring up various results for annotated pages related to E. coli proteins that have been detected in 2D page gels. So this is a page devoted to one particular protein called DHE4. We can call this up, and we can see that there are a couple of 2D page gels in this repository that have been annotated for having presence of this protein. We can pick up one of these pages, and we can see exactly which spot on this 2D gel has been annotated as being the spot corresponding to that protein. So these resources are not as easy to use as the gene expression ones. Images are generally not as easy to work with as strings, so easier to work with a sequence coming off a sequencer than an image of a gel, but there are resources available for this. Mass spectrometry, I mentioned, is probably, most people would say, the more promising technology for getting to really high throughput protein studies. Unfortunately, we don't have as good resources online to just manually go in and demonstrate the kinds of things available in mass spec resources. There are now some repositories of mass spectra, but you can't just go and browse them and play around with them online. You have to install specialized mass spectra viewers and download these fairly large data files, so it's not something that would make a good demo for now. But if any of you actually want to work with mass spectra, the resources are out there, so you can get these. I'm just not going to be demonstrating them here. But basically what this should be giving you an idea of is the fact that we have some resources for looking in a fairly large scale at protein expression. They're just not as good as the resources for looking at gene expressions, that is RNA expression, for many of these kinds of questions. Right, so there is one more thing I wanted to at least mention here fairly briefly. We'll go into this in much more detail next week, but one final resource that is worth mentioning is the gene ontology. So the gene ontology, for our purposes this week, is useful because it gives us a way of looking at expression at even the subcellular level. And what I'm going to show you here is just briefly how we can get to that information through a tool called Amigo. And I actually find it easier to use an older version of Amigo, so click on this, Amigo 1.8. This gives us ways of looking at particular genes or particular locations within a cell to see what genes are active in these. I don't have time today to explain in any great depth how this works, but in the basic sense, what we can do is type in a gene, let's say. So I'll type in this gene, ABC4, say that I'm looking for a gene, submit a query, and this will give us information on this ABC4 gene. We can see that maybe we're looking for ABC4 in Arabidopsis thaliana. You can see that that's a particular gene here, and we can get information about where, what this gene does, where it's located, and about terms associated with it. Because what the gene ontology really is a collection of terms. So annotate what's called an ontology, which is the way of organizing information, similar to that anatomy browser I was showing you before. And what we can see is that this has a fair number of terms related to what's called the cellular component, which is where this gene is found subcellularly. So we can see that this is annotated as being found in a chloroplast. In particular, it's found in the membrane of a chloroplast. And we could learn all about this gene or any other gene annotated here with respect to that category. We'll note that this data set, this Go repository, is actually more often used for this biological process category, a set of terms that relate to biological functions. We'll see a lot more about biological functions next week. but these terms are things that we would often use to kind of figure out which things are located in which pieces of information. So we could pick on, let's say, the chloroplast membrane term here, and we could look at things from the point of view of a particular localization and figure out if we are looking at, if we are interested in looking at that localization, find out about all the characteristics of that localization, so figure out exactly what a chloroplast membrane is, how it's related to various other categorizations of places within a cell, and we could, of course, look up all the other genes that are annotated as being found in chloroplast membranes that maybe interact with this gene of interest to us. So as I say, I'll be saying a lot more about the gene ontology next week. It's actually much more relevant when we cover the issues of systems biology, so how genes function in networks and pathways, but I just wanted to mention it today because it happens to be a good place to learn about subcellular patterns of protein expression. So I think we are out of time here. Any last questions before we break? Okay, if anyone hasn't given me your homework, please make sure we have that.